Hello, in this video we will look at the lipolysis. The lipo means the fat and lipid, and lysis means the breakdown. So the degradation of fats, and that is fat is present in the adipose tissue. So the storage of the fat will that convert into the glycerol and free fatty acid is known as the lipolysis. So it is a complex mechanism to understand, we will discuss. So the breakdown of triglyceride into fatty acid. Triglyceride mean the triacyl glycerol mean the three fatty acid chain contained with the glycerol. So here is the glycerol and free fatty acid will be converted after the triglyceride. The triglyceride is the importance is that is the storage form. So the breakdown of triglyceride into fatty acid. So here is the triglyceride which that is converted into the glycerol and fatty acid. So this means the hydrolysis will be occur and convert the triglycerol and fatty acid, and this is a cause due to the glucose level low into the blood due to the fasted state. So this mechanism play in the fasting state. So the enzyme involved in the lipolysis is the ATGL, adipose triglycerides lipase, which that is hydrolysis of triacylglycerol to diacylglycerol, and the HSL, which that is the hormone sensitive lipase, catalysis of diacylglycerol to monoacylglycerol. While this is hormone sensitive, it means the glucagon sense as after the sensing of the glucagon or epinephrine, which that is due to the sympathetic nervous system during the fasted state, which that is produce this HSL. While the MGL is the monoacylglycerol lipase, and MAG is the glycerol, basically is the monoacylglycerol, which that is converted into the glycerol. And the perilipin is the protein which that is perilipin 1A is regulates the lipolysis pathway. So here is the pancreas during the fasted state. So here is the pancreas which that is produce the glucagon and inhibit the insulin production from the beta cell, but the alpha cell produce. Here is the group of tissue is known as the acini or islet of Langerhans, which that produce glucagon, and this glucagon will stimulate the lipolysis by the binding of glucagon or epinephrine with the adipose tissue, adipocyte basically. So that's adipocyte. It means the breakdown or degradation of the fat from the adipose tissue due to the fasting state, producing of the glucagon, while the inhibition of the insulin, because insulin is stimulated due to the fat state. So remember about that. Why? Because the increase of the blood sugar will dispose into the cells. So here is the adipocytes, which that is basically adipocytes contain a receptor. So this I am drawing the plasma membrane of the adipocyte for understanding and the breakdown or degradation of the lipids or fat. So here is the plasma membrane of the adipocytes, which contain a basically G-coupled protein receptor. This G-coupled protein receptor is basically is the integral protein. So basically here is the epinephrine or glucagon basically, or epinephrine will be bind basically epinephrine. So this is the ligand and it is the primary signal, and after the primary signal binding to change the intracellular conformation. So here is the 3 monomeric protein containing G protein. This protein is contain alpha unit, beta and gamma. This alpha unit contain a GDP in the form of inactive state, while during the activation due to the binding of the epinephrine, ATP will convert into the ADP and phosphorylase, and this phosphorylase is due to the phosphokinase enzyme. So after this phosphorylation, it will alpha subunit or alpha monomer will be separate from the beta and gamma. So the beta and gamma will remain that place while the alpha subunit will be released or detached. After the detachment, it is converted into the GDP from GTP from the GDP. So it is the active form will lead to enter into the toward the adenylyl cyclase or adenylyl cyclase. So this adenylyl cyclase is alpha subunit attached from this place, and it will be ATP used to convert the cyclic AMP. And cyclic AMP, that is why it is enzyme is known as a cyclase, because it is convert the ATP into the cyclic AMP. After this, the inactive pKa, that is the protein, will convert by the stimulation of the cyclic AMP to convert the pKa active by the phosphorylation due to the kinase enzyme, kinase activity. And this active formation which convert into the HSL hormone sensitive lipase, which convert phosphorylation to phosphate group attached with the hormone sensitive lipase enzyme. So this hormone sensitive lipase enzyme will play a different function. So here is the I am drawing the liposome. This liposome contain lipids inside. So here is the perilipin 1A is present, the three monomers which contain a CGI also attached. So the pKa, which that is the active, and this active pKa phosphorylase it, the perilipin 1A. After the both monomers will be phosphorylazed and detached. So the detachment of the CGI with the phosphorylation, after the phosphorylation, it will be detached and it will be stimulate or phosphorylase into the ATGL. So the ATGL is the adipose triglycerides lipase. This triglyceride lipase is basically convert the triacylglycerol into the diacylglycerol. So we will discuss. So first of all, the HSL hormone sensitive lipase will be bound with the HSL after the phosphorylation with the perilipin protein subunits, three subunits. So here is the ATGL, which that is basically the triacylglycerol will convert, that is known as the adipose triacylglycerides lipase, which that is convert into the triglyceride to the diacylglycerol. And after the diacylglycerol will convert into the monoacylglycerol. After the monoacylglycerol will convert due to the MAGL. MAGL is the monoacylglycerol lipase enzyme. So after this conversion, free fatty acid will be released out, and releasing out, it means the free fatty acid. So the HSL is release the monoacylglycerol and convert into that place. So you can imagine that. While on the other hand, important thing is that here is the pKa, which that is stimulate the AKAP. And after the stimulation of AKAP, which that is for your understanding, so basically is the phosphorylation by the pKa. So the pKa is bind with the AKAP is and bind. It means the 4 cyclic AMP will be bind to stimulate pKa. It means the 5 pKa stimulation for each signal is, it means that 20 cyclic AMP is required. So it means the 4 cyclic AMP required the 1 pKa activation. So it is activate and 1 pKa activate to release the 1 triacylglycerol into the free fatty acid and glycerol. So remember about that. On the other hand, here we will discuss after the transport the fatty acid into the blood and into the organ, and what is that during the fasted state. So important thing is that it is during the fasted state. So here is the blood vessel. This blood vessel is basically is the part of circulatory system which that is used for the movement and transport the different material to another cells. So that is why it is known as the circulatory system, and absorb the nutrients where the need. For example, the adipose tissue absorption into the blood. So here is the absorption, and here is the absorption into the tissue. For example, the liver, when absorb the free fatty acid into the liver as well as the triacylglycerol, these free fatty acid and glycerol will convert into the acetyl CoA by the mechanism we will not understand here, and ketones and very low density lipoprotein and cholesterol. This is the acetyl CoA will produce the ATP when go into the mitochondria, basically is in the mitochondria, while keto acid is produce ketones. So the ketones in the form of keto acid, beta hydroxybutyrate and acetoacetate. So when this during the fasted state, if we will not care with the fasted state or some mistake, so ketoacidosis can be occur, or diabetic ketoacidosis if we are diabetes. So here we can check that by the ketometer to check during the fasted state, while it is also go into the tissue free fatty acid and convert into the acetyl CoA in the mitochondria and produce ATP. And one more thing is important, that the ketone is used in the brain, while the fat is not used in the brain, while the ketones used into the brain. So the brain is prefer more ketones, and muscle cells also. So here is the important thing in summary is the glycogenolysis is the degradation of glycogen before degradation of adipose tissue. So the glycogen degradation is the first, so remember about that. And lipogenesis is the inhibition and lipolysis triggered by the sympathetic nervous system activation to release glucagon. So I hope you make sense about this video, lipolysis. So next we will discuss about the another glycogenesis.